not a computer person. So, Microbes and Bees. 2008, Dean and I went out to Arizona to work with Dee Lesby, and on that trip, we met Dee's bees, decided to get married, went back to the desert like 6 weeks later and got married. And we were driving back from the wedding, the very tiny little wedding under Kitt Peak. And the truck ride home from the wedding was like an hour and a half back to Dee's place, and we were really tired. We had hived like 7 swarms that morning, and finished our like wedding clothes and like driven to the desert and like met up with these Minutemen that ended up being our witnesses, and it was like a really, really great adventure, but we were really tired and trying to make small talk on the way home. And Dean happened to mention an article that he read, a little thing in the classroom section of ABJ, Dave Mendes writing to Jerry Hayes asking for a recipe for pollen substitute. And the answer from Jerry was like, well, bees don't really pollinate bee bread. And there's something like 20 different microbes involved in the fermentation of the pollen into the bee bread, and yeast and molds and bacteria. And as soon as I heard that, it was like a click went off in my head, and I thought, oh my God, I think the microbes might be really important. Why is nobody talking about this? And it's like, I just had a vision, and we instantly, like that night, we got on the computer and we started like looking for stuff, and it basically had months of frenzied activity of just looking at everything we could find on bees and microbes and humans and microbes. So this talk is kind of like a brief synthesis of a bunch of the things that we've learned since then. And we're basically more convinced than ever that the microbes are fundamental to the health of the bees. So I like to start first by getting everybody into their imaginations, because some of this stuff, it's kind of hard to picture if you don't have kind of a real life way of thinking about it. So I like to think about if we were aliens and we were, kind of floating in space and we found this planet, what would we really know about it? Like, would you, if you were an alien, what would you know? But we're not aliens, so we know something about our planet. We know that this is North America, this is South America. We have some ideas about life forms that occupy the planet, the landforms. We have ideas about things that occupy the ocean. We have things that we know about different cultures and how cultures migrate, or how economics works in our different areas, or just, all of us have different ideas of these things, but we have some ideas that we can make pictures in our head of how things interrelate and move around and affect each other. If we get a little closer in, aerial shot of Philly. Any of you from this area probably have even more information about what goes on in the city, your neighborhoods, how the businesses work and interact, all sorts of things about how things affect each other and interrelate. So we can use these kind of pictures or imaginings to start to think about the microbes, because the microbes are functioning in many ways similar to other life forms that are bigger that we already know about. It's just they might move about differently, and they might have different functions, but basically, if we think about populations and movement and economics, anything that has to do with systems interrelating, we can imagine the microbes better. So we're going to talk mostly about yeast, bacteria, and fungi. Viruses are in here, but I'm not going to talk about them too much, because that's a whole other complex side issue. But we include them as part of the invisible life forms in the hive, even though they're kind of on that edge of life, not life. So all living things need a suitable environment, some means of sustenance, right? You need a place to live that suits your biology. So if it's too hot or too cold, or too acidic, or too abrasive, or you can't get good shelter there, like there's things just on an environmental level that every life form has to think about before it sets down and can propagate itself. Sustenance, you need some way to like either find food, grow food, make food. Everything needs to eat. You also need some kind of survival skills, and different skills will be different for each kind of living thing, but everybody needs to have a skill to stay alive. And on the group level, on the individual level, you don't need a means of reproduction, because you could just decide, this is it for my line. But on a colony or group level, every group needs a means of reproduction, or the species can't go on. In the world, in the planet that we live on, microbes are the necessary foundation of larger life forms. So basically, on the invisible level, the microbes are supporting all the life above them. The microbes are everywhere. They can live in thermal vents in the ocean. They can live in very arid climates. They can live in spore form and live for many, many, many years and then reactivate when the conditions are right. They can live in very dry areas. They can live without air. They can live without light. Almost any place on the planet or in any organism, there are places where microbes live. They also make up the bulk of life on the planet. And one of the most important things is that they occupy and adapt to advantageous niches. So seeking out environments that will support them. And these niches are really important because many of the microbes have co-evolved with other living creatures, and they help each other. So basically a creature will offer up, say in a gut, here, you can come live here, and you can do these tasks for me, and I'll give you a place to hang out. And sometimes it's just, I'll give you a place to hang out, and sometimes it's actual activities are happening between the organisms that are doing more than just giving space, but helping each other with metabolism or any of the other life things that we talked about. They're nature's ultimate recyclers. I think if the bacteria and the yeast and the fungi disappeared from the planet could go on about 2 days before everything would seize up. Because they're recycling everything as things grow and die. The microbes are transforming the dead matter back into living matter. Just that's a process that's constantly going on like large and tiny levels. And they make the continuation of life possible. They prepared the planet for all other life. They were our first life forms, and they populated the planet. They support all the larger life forms and live in our cells as mitochondrial DNA. So all of us have parts of ancient bacteria that has basically become part of our cells. I like to think the meek shall inherit the earth. It's kind of like a dust-to-dust thing, like everything started as a microbe, and basically, on some level, everything will probably finish as a microbe, both individually and on a larger scale. So microbes, like all life forms, compete for resources. They utilize unutilized food and waste, and individually, they can't really do a lot as an individual, but as a colony, almost like a bee colony, they can act as a larger life form. So they can create biofilms, they can create, the bacteria can create biofilms and act almost as a creature. The fungi can create mycelium and act as a creature. They can do a lot of interesting things. There's a great book by, forgetting his name, scientist guy. Microcosm. Yeah, Carl Zimmer, yeah. He has some great books on microbes. The Microcosm book was really wonderful, and there's also a fungal guy, a mycelium guy, the mycelium running guy. Yeah, he's interesting too, but both of those, especially the Microcosm book, I would really recommend if you're interested, especially in bacteria. Very fun to read. So the microbes are always balancing. And they require competitive cooperation and exist in complex adaptive cultures or systems. So just like we think about, like say the city of Philadelphia, you have, the city has needs, food has to come in, waste has to go out, commerce has to be done. There's all kinds of systems that are interacting with each other, and kind of almost like a stretchy balloon type structure. You push on one end and something pokes out on the other end. The microbes are acting that way too. If one microbe takes over and uses up all the resources, basically that can be an end for that particular species in that area. So there's a lot of like give and take between the cultures and between the organisms so that everybody can kind of go on and exist and nobody completely wipes the other one out. And sometimes that does happen, but for the most part everybody has to act in a kind of competitive, cooperative way. So this little drawing I like to sort of illustrate just on a kind of conceptual level. You could see the spirals as individuals or you could see them as groups, but basically things have to eat. In the eating, there's metabolizing, and they're giving off wasteful stuff, or toxic wasteful stuff, or useful stuff, depending on who you are. So one organism's waste or toxic output could be another organism's goodies. And this kind of spiraling and taking in and giving out is going on all the time on every level. So locally and globally, microbes are the unseen, unappreciated masses. And I like to think of them as sort of like the janitors of the world that, you know, janitors, cleaners, anybody that's occupying sort of what we think of as like a lowly task, they perform so many needed, the tasks they're doing are so necessary. Like that if all the janitors suddenly disappeared from the planet, all the trash pickers, all the people that work at the dumps, all the people that move waste around, anything on that level, like we would be overrun in like a day. So there's many people in the world that are doing jobs that are not really even noticed. And the microbes are kind of in that role for me. It's like they're performing all these tasks in organisms, in the soil, in bodies, in guts, on skin, in the air, like they're everywhere and they're doing all these jobs, but they're invisible and unappreciated. The vital tasks that they're performing affect the bottom line for many organisms. If the microbes weren't there doing the job, the cost to the organism might be much greater than it could afford. So I think of microbes as sort of a way that organisms outsource work. Like in the gut says, okay microbes, come on, you can move in here. And while you're here eating my waste or metabolizing things to be put back into my body, you're going to get the food you eat, and meanwhile I'm going to get the vitamins I need. Or I'm going to get the nutrients broken down into a way that my body can reabsorb them and use them. So there's, if the microbes weren't there doing the job, the organism would have to figure out another way to make that stuff happen, and it's not clear that it would happen economically or that it could happen at all. The microbes also fill in gaps in the system wherever they may be. There's always an opportunity for something to move in. An interesting example is on our skin. Our skin is covered with streptococcus bacteria, and, staph bacteria. That staph holds off, it basically holds space. It's filling in a niche to hold space against invasion of fungals. And if you strip off too much of that staph, you could get a fungal infection. So basically, the one bacteria is holding space so invaders can't move in. Now, if you cut yourself and that staph gets into your bloodstream, that can be really bad. So in one place, the staph's performing a really important job, in another place, it could really cause trouble. So stripping away microbes, I mean, one of my pet peeves is like incredibly prolific use of antimicrobial gels. Like everywhere I go now, there's like a big old, you guys seen them? Like you go to the bank and there's a big old antimicrobial pump. Like everything's like get rid of the microbes. Soap and water is fine for cleaning. You don't need to, and that will not strip the staph off your skin. But antimicrobials used over and over again, you're like opening up niches for other virulent things to move in. So in our quest to be clean, sometimes we're actually making things worse for ourselves. And for the most part, the microbes are beneficial or innocuous when the environment's stable. For how many microbes there are in the world, the amount of problems that they cause is very small. And many of them are working in teams with plants and animals to basically have the organisms function the way they do. Now, for the bees. The bee-related microbes have co-evolved with bees and plants over millions of years, and some of these microbes are unique to the plants and the animals. Some of them don't live anywhere else on the planet, but in the place they live in the plant, in the animal. They're an integral part of the honeybee anatomy and biology, and must be included as vital members of the colony, and rarely cause disease. Plants are very important to look at while we're talking about the microbes because the bees are entirely dependent on the plants for their food source. The honeybees have a very wide diet in terms of different kinds of pollens and nectars, but that's all they eat is pollen and nectar. So we have to think about plants too. Plant-associated microbes recycle nourishment through decomposition, so they're working on the soil level to release nutrients that the plants can reabsorb. They feed root systems, and many times the plants are feeding sugars out through their roots to support the microbes, so that the microbes can then exist to feed back to the plants. So we have this very nice cooperation going on in the soil. The plants the microbes can be defended against by toxins produced by plants. So the plant, some microbes might be helping the plant on the root level, but invading on another level. And a lot of what resins and toxins are in plants are to keep that from happening, to keep them from being eaten or infected. But the bees can appropriate these toxins for their own defense. And that defense, again, can be physical or microbial. And that's in the propolis that they make from the resins. The honeybee colony we think of it as a simple system with very complex needs. So very few inputs. We have pollen, nectar, water, and plant resins. But from those few things, the bees have to create a nest structure. They have to be able to make comb. They have to be able to rear brood, preserve their food, and they have to be able to mount a physical defense and a microbial defense. And all of that's happening through those few inputs, and the microbes are basically the facilitators. We have our bees with our bee-associated microbes. It's kind of like a nice little way of thinking of a more whole picture. The plants are contributing pollen, propolis, and nectar, and with those things, they're associated microbes. The bees have the associated microbes in their guts, and between the bee microbes and the plant microbes and the food inputs, the bees are able to provide for themselves. Then there's microbes in water. The bees are consuming water, and also the water is going to nourish the plants. At the plant base, we have microbes that are nourishing the roots. So there's there's this constant kind of fluid trading back and forth of resources. And then in turn, the bees pollinate the plants and let them perpetuate. So the three things that probably are the most important are the microbes being integral to honeybee defense against disease, and a lot of that's happening in their guts and in their food supply. The production of the food supply through the bee bread, and the digestion and assimilation of nutrients in the gut. And again, we have our colony cast of characters, including the microbes. Now, one of the most important places where the microbes live are in the gut of the bees. And has anybody here been following the Human Microbiome Project or learning anything about human gut microbes? Anybody? Okay. So we have guts too, and a lot of the research on humans, we have a lot more information about human gut microbes right now than we do about bee gut microbes. But any organism with an inside has a gut. And so we know that other animals, termites and aphids, those are two other insects that have guts that are very dependent on gut microbes. So as the information starts to build in the bee arena, I think it'll probably come out that the gut is one of the most important places in the bee, for the bee's health and for their well-being. So you need a gut because you need a place for nutrition to come in and waste to go out. So anything that has more than a cell has to have a place for nutrition to come in and waste to go out. And in the gut, the microbes are controlling fundamental aspects of the organism's function. The gut is vital to the health of the organism, and microbes are critical to the gut function. So here we have our Honeybee Anatomy. Talk about the gut in a minute, but we also have the flight muscles, or the striped bands, and the hypopharyngeal gland and the mandibular gland. I'm talking about these because when we talk about nutrition, these will be really important. The two glands are the places in the bee's body where the food is being manufactured for the young larvae. So that glandular development has to be good for the food supply to be good. And the wings control both the thermoregulation of the hive and the foraging. So without good, strong flight muscles, you're going to run into problems in terms of bees being able to forage effectively or control the temperatures in the hive. And good development of both those muscles and glands are highly dependent on the nutrition that the larvae gets. The yellow, kind of balloon shape, that's the honey stomach. We have lactic acid bacteria that lives in the honey stomach that I think some of the species in there are unique to the bee. They don't live anywhere else on the planet. And that lactic acid bacteria is fundamental for the inoculation of the pollen that we're going to talk about in a little bit. Moving on, the little bright blue valve, that separates the honey stomach from the digestive stomach. Then the midgut is the orange area. That's constantly shedding its cell lining, so there's not a lot of microbial activity in there. But the two places where there's a lot of activity are the ileum, which is the bright green thin part, and the rectum, which is the bright blue part. And it's suspected that most of the digestion and assimilation of nutrients is going on in the ileum. It's a really important part of the bee. Now the microbes that live in the gut, all the way from the honey stomach back to the rectum, many of them are the same species. There's about 8 species of microbes that they found that make up about 95% of the gut. And these are bacteria. And many of them, I think all of them pretty much are unique to the bee, that they've evolved inside the bee to their own special strain. And the ileum is where they're particularly rich. So these are some of the microbes that have been identified with Nancy Moran's lab. I'll talk about her a little bit later, and Dean will be talking about her also, but she was heading up a lab at Yale, and she's since moved her lab to the University of Texas. But her lab is doing some of very fundamental research on what exactly are the microbes associated with the bee gut. And her previous research was gut microbes in aphids. So she's had a long career of looking at gut microbes, and now she's turned her attention to the honeybee. So we have many, many questions for her. We ask her a lot of stuff, and right now they're working on really fundamental, like building blocks of the system. So all the interesting questions we have we'll have to wait probably for a bunch more years, but meanwhile they've done the fundamental identification of the gut microbes, at least the 95% that make up the gut. And it's interesting because the different areas of the gut have different, we talked in the beginning about environments, that you need to have an environment that supports the organism. So every place in the gut has a slightly different pH. The acidity in the honey stomach, in the midgut, the ileum, and the rectum, they're all different. So they tend to support different populations of these 8 microbes. They tend to exist in different densities and quantities, depending on which part of the gut they're in. Yeah.
Talk about the names of the first 3 out there.
Oh, right. The Snodgrassella was named after Snodgrass, who did the first very intricate drawings of the anatomy of the bee, the internal anatomy. What's his first name? Snodgrass guy. He's famous. Right, doesn't need a last name because he has a great first. Doesn't need a first name because he has a great last name, you just say. Frischella is named after Carl von Frisch, who studied the dancing bees, the bee dance. And Gilliamella, that's our favorite. The Gilliamella apicola. Martha Gilliam was a researcher in Tucson at the Tucson Bee Lab. She was there from the 70s through the 90s, and she was the person who did the bulk of the fundamental research on honeybee microbes. She was basically the first person we found when we were researching, and we had an opportunity to go to Tucson a few years ago with a scanner and go to the library and scan all of the documents that, all of the scientific papers that she'd published throughout her career, and those are all available on our website for anybody that wants to just totally nerd out and read old studies. They're actually really fascinating. But she's, you know, almost any paper you read now on gut microbes or any kind of microbes in bees will reference her work. Because she was, there's some work that goes back to like the turn of the last century. I think I found something from like 1906 that first started talking about bacteria in bees. But the bulk of the work came from Martha Gilliam. So they honored her with naming one of the gut microbes after her. This is a slide from Nancy's lab. And again, the ileum. The green, skinny thing. Picture that cut cross-section. This is some of what they see there, and the ileum, basically, it has these 6 extremely deep folds, and lining the folds are some of these bacteria that don't exist anywhere else on the planet. And not only that, but the bacteria have evolved with the lining of the gut so that they can't be separated from the gut. If you try to take those bacteria out and culture them, they die. They literally can't live without their attachment to the gut. So it's sort of like they are microbes, and they do have some independent status because they have separate DNA from the lining of the gut, but they can't be separated. They've just evolved to be like one organism. So one interesting thing is, well, how does the organism get inoculated? You know, we're like, as humans, we're born sterile. When we emerge from the womb, we do not have microbes in or on our bodies. And when we get inoculated, the first thing that happens is we either are born through a birth canal, like a vaginal birth, where we get one set of microbes, both through vaginal mucus, through our noses and mouths, our guts get inoculated, or the skin gets inoculated also with bacteria. And if we're born with a cesarean birth, those microbes are going to be really different. Breast milk versus bottle feeding, the microbial cultures are different in the gut. And there's talk now of ways to basically get infants up to speed. I know I found a website for women that were facing cesarean births, and they were basically going to harvest vaginal mucus right before their babies were born so they could feed the babies and get the babies inoculated. And there's other people that are looking now at prebiotics and probiotics for infant formula, so that people that can't breastfeed their babies can get their babies inoculated. And with the bees, it's the same thing. When the bees in the larval state, it's been fed a really rich diet of bee milk, and in that bee milk we have some of the lactic acid bacteria from the honey stomach that help hold space in the larvae against like invasions of American foul brood. There's resistance there between those bacteria, the lactic acid bacteria and the foul brood bacteria. But when the larvae reaches its state where it's going to be capped and pupate, all that food that is ingested over that 6-day larval period, none of it's been excreted until the very end before it pupates. It poops one tiny little poop out. And with the poop comes its gut lining. So when it goes into the pupal state, it's sterile again. Now when that bee emerges, she doesn't have any gut microbes. So the first thing that happens is she chews her way through the wax, and she gets some microbes from the colony in her gut through that chewing. Then there's feeding between the bees, the trophallaxis. As she's emerging, the bees will come and feed her, and then they'll feed her after she comes out. Some microbes are being ingested that way also. Then she does her hive cleaning between 1 and 4 days. She's also going to consume pollen and or bee bread and nectar and or honey. So some microbes are going to come in that way as well. And then also through grooming. Now the hive cleaning is, to me, the most interesting because Dean will go into this more in his talk, but that period of day 1 to 4 is when the gut of the bee is becoming inoculated. And at the beginning of the inoculation, there's a lot of different microbes that don't necessarily end up being the final populations, but by day 4 or day 6, the bees basically have their microbes that they're going to go through their life with. That's also the time that they're cleaning the hive and ingesting any kind of residues in the hive. And that's the period where I'm suspicious that the poop ingestion is happening. They also do a lot of grooming, and there's probably fewer microbes that are being ingested that way because the bees don't carry a lot of microbes on their exoskeletons. Yeah? Oh yeah. You'll often hear it said that bees don't defecate in the hive. One day, a couple years ago, Dean was outside and a bee pooped, and he got some poop on his arm, and he took a little piece of straw, just picked a piece of straw off the ground and made a little fishing line, and dangled it over the top bars. And within a few seconds, a couple of bees came right up and cleaned that straw immediately. We also have an observation hive, and we spend a lot of times at markets with it, and we've seen bees poop in the hive, and almost instantly another bee will clean it up so quickly that you don't even know that it happened. So,
they don't fly immediately.
Right, and the bees don't do their orientation flights till they're like 2 or 3 weeks old. So, what's happening during those first few weeks of their life? They're ingesting a lot of food, especially the nurse bees, ingesting huge amounts of food to feed the young larvae. And I don't know, I'm suspicious that I don't know that I believe anymore that bees don't poop in the hive. First, because I've seen it, but I'm almost wondering if instead of an accident, it's something that actually happens as part of the inoculation of the young.
That's how termites get inoculated.
Right, Steve just said that's how termites get inoculated, through eating other termites' feces. So again, we have our potty mouth slider, biological warfare. Now the bee bread. While the bees don't get all of their gut microbes from the bee bread is a really important place for microbial activity. Again, bees don't eat fresh pollen, they eat fermented pollen. And during the fermentation process, unique microbes, unique nutrients are being produced that the bees can't get anywhere else in their diet. And that includes several things. So here's a little flow chart of the inoculation and fermentation process. First, we have our pollen and its resident microbes, and most of them are yeasts and fungi. And the bees collect the pollen, and immediately they moisten it with saliva from their honey stomach. And that saliva includes the unique lactic acid bacteria that we talked about earlier. And almost instantly, a fermentation process begins. So we have the yeast and the fungi and bacteria all together start some microbial activity. And the bees bring the pollen back and store it in the comb. And there's a very huge metabolic activity over the next 12 hours. There's a lot of stuff going on where, sort of like we talked about the microbes in the beginning of the gut inoculation, not being the microbes that they end up with, but possibly setting the stage for the further microbial activity. So the first thing that happens is, through that intense metabolic activity, we have a lot of lactic acid bacteria being produced, and that sets the stage for fostering the lactobacillus that end up being one of the predominant microbes in the bee bread. And that's like a pollen pickle, or like a fermented pickle. That lactobacillus is what preserves the pollen and also enhances its nutrients. And you end up with bee bread after about 2 weeks. That microbial activity finally settles down. But in the meantime, at any point in that flow, the pollen has already been at least partially fermented. So again, we end up with the pollen pickle. And in the pollen pickle, that bee bread now has antibiotics, it has organic acids, vitamins, enhanced protein, fatty acids, sterols, and enzymes. And one of the sterols that it has is something called 24-methylene cholesterol that seems to be fundamental for the rearing of the brood. And bees can store that in their bodies for about 2 brood cycles. So if they don't have access to real pollen, I think they can raise about 2 rounds of brood before their bodies get depleted. So it's sort of like a nursing mom. If you're nursing your baby and you literally have no nutrition, you can draw from your body, and it's probably going to come out from your bones and your teeth and your organs. Those nutrients are going to leach out to be feeding your baby. But at a certain point, you're going to run out. You just won't have the resources to keep going. And if you feed pollen substitute or supplement, none of those things work unless you have at least 10% real pollen in there. The bees need to have at least some real pollen to keep the metabolic activity going in the right direction. So, nectar and microbes in honey. Again, we've got the unique lactic acid bacteria that live in the honey stomach, and as the bees are gathering the nectar, those microbes, those bacteria are mingling with the nectar. And it seems that there's some evidence that these lactic acid bacteria can vary with nectar sources. So increase or decrease depending on what kind of nectar the bees are gathering. And those bacteria protect the nectar from mold and yeast fermentation. So it's a way to kind of keep the nectar stable until the bees can get it processed into honey. Honey has antimicrobial qualities, and it can also detoxify natural substances in plants. The bees can use it as a way to keep toxins from developing in the hive. Also, diluted honey can support microbial growth. So if the bees need to, grow something, like mead, they can dilute the honey and promote microbial activity. And honey is also a part of the bee bread process, because at the very end, when the bees finish the bee bread, they cap it off with a tiny little glaze of honey to further the preservation. So this is a little honeybee nutrition cycle. You can start anywhere in the cycle, but we'll start with the foragers. You have a forager gathering pollen and nectar, but for this we're going to focus on the pollen. Forager, as soon as she inoculates the pollen, that microbial activity starts. She brings it back to the hive, it gets made into bee bread. Now, your young nurse bees are consuming huge amounts of the bee bread to make the milk to feed the young. So for the 6-day larval process, those larvae are basically swimming in brood food. And this nutrition that they're getting in that 6-day larval period is going to determine their biology, their health, their muscular and glandular development. Everything that the bee is going to become that's not genetic is basically in that bee bread, or in the bee milk that's coming from the bee bread. So after 6 days of swimming in the food, the bee will create her cocoon. She'll emerge, and within 4 days of her emergence, she's back feeding the next round of young larvae. So you have a very tight cycle there of resources, inputs being turned into resources for the young larvae, and then that resulting bee, whatever nutrition she got, is going to determine her health and her ability to go out there and forage and create healthy food for the next generation. So we have a really big feedback loop there. If something goes wrong with the pollen, or the bees don't have properly fermented pollen, or the pollen is somehow tainted or not adequately nutritious, you can have a very quick spiral downwards in the health of the hive. So we'll talk a little bit about microbes and disease. I love this quote. It's helpful to remember that in nature, friend and foe, cause and effect are not always distinguishable. I like to think about microbes taking up niches. We talked a little bit about the skin. I think about things that we think of as good or as bad, like positive, negative. In the microbial world, I don't think things are that clear. I don't think they're even that clear in the world that we inhabit. But one example I like to think about is if you live in a neighborhood that doesn't have gangs, you might have ideas about gangs and think, oh, gangs are bad. But what if you're in a neighborhood that does have gangs, and the gang that was in your neighborhood is actually protecting you on some level? And suddenly something happens, the gang goes down, now there's an open niche. Who moves in and takes over? And we can see this on a global level with world politics. We can see it on a personal level. There's, everything's sort of holding space. What happens if that undesirable thing suddenly changes? What takes its place? And I guess it's sort of the old saying, the devil that you know, the devil that you don't know. There's a lot of examples we can think of for parallels there. But in our quest to sort of deal with microbes, I guess when Pasteur, just a little over 100 years ago, discovered the first bacteria, and he was working in a realm of bacteria that were causing a lot of harm to the silk industry and the wine industries in France. So once people figured out that these bacteria were the root of the problem, for a long time we've had kind of an attitude of the microbes are bad, we have to deal with them. But until very recently, there hasn't been a lot of information about how the microbes are actually doing many, many, many vital tasks. So I guess I'm thinking in our quest to keep our bees healthy, there's a lot of emphasis on disease and what are we supposed to do to deal with these diseases without really understanding the repercussions of those efforts. So microbial imbalance expresses in disease. We've got the common ones that we're used to are American foul brood, European foul brood, and chalk brood. We've got 2 bacteria and a fungus. Nosema, which is nobody can ever really definitively decide what to call it, but right now I think it's Microsporidium that's either a fungus or related to a fungus. It's in some weird camp. And then there's the viruses that we all hear about. And viruses, I put a ton of question marks because, number one, I don't really feel prepared to talk about them in detail, and number two, I think that they're quite mysterious, and I don't know that anybody clearly understands what's going on with the viruses. You can have a bee that has a high level of virus that doesn't exhibit bad health or adverse effects, and then you can have bees with low levels, and this is for the nosema also, exhibiting low levels of nosema or viruses and still succumb. So I put those separate because that's a whole other topic that I'm not really prepared to go into. This chart is kind of an attempt to illustrate pressures and what happens when we attempt to deal with those pressures. The molds are in green, the bacteria are in red, and the yeast are in yellow. And within the molds, the yeast, and the bacteria, just say within the bacterial community, there's competition. Different bacteria are holding space, other bacteria want to get in there and take the space, so there's kind of this competitive, this vying for resources. For one example, like the two foul broods tend to be antagonistic to each other. So if one's holding space, the other one's less likely to take over. Within the molds, there's competition, and within the yeast, there's competition. But not only within those families, there's competition between the different families. So you have yeast competing with molds for space, and bacteria competing with molds, bacteria competing with yeast, so that competition is going on. Now, say we want to deal with some bacteria. Say we're afraid that our hive might have a foul brood, and we want to deal with it with an antibiotic. Well, we can use the antibiotic to put pressure on that foul brood bacteria. But at the same time, we're also putting pressure on all the other bacteria in the hive. So now we've created, wanting to deal with one imbalance, we've created another imbalance and opened up niches now for, say, yeast or molds to take over. And there are, for all these things on here on this chart, there are laboratory evidence that, that these things do have causes and effects, that treating your bees with terramycin definitely impacts the bacterial communities in the bee and also opens up space for yeasts. Same with feeding high fructose corn syrup or sugar. You're altering the environment in the gut of the bee, and so you're kind of changing the niche opportunity for the different bacteria and yeast and molds that live in the bee. Fungicides are probably, and organic acids. Organic acids, in the lab, we know that if you have organic acids, looking at organic acids in bee bread, say, the effect of the organic acids on the bee bread, and this is the same too for the high fructose corn syrup. Basically, you can alter the molds, and molds are like a really important part of the bee bread. That's where a lot of the antibiotic activity is coming from, the molds and the bacteria. If you alter that mold, the mold communities, some molds now will grow faster and others will grow slower. So you end up with like a different substance than you started out with. And that's not to say that like nobody should use an organic acid or that an organic acid is going to kill your community, but these are all unknowns because a lot of this stuff they're looking at in laboratories and it's not clear in the field the exact effect that all these things are having. But we know, at least from the laboratory experiments, that organic acids, sugar feeding, antibiotics, all of these can have an effect on the communities in the hive, the microbial communities. Fungicides, the fungicides and the terramycin I colored in orange because those two are probably two of the most important ones. And I probably should have colored the fumagil in orange also because that has a really big and can exert a big pressure on the bacteria. With the fungicides especially, I don't know how many of you have bees near intense agriculture, but fungicides are legal to be sprayed on open blooms because they don't affect the health of an adult bee. But until recently, nobody was looking at the effect on the food that the bees have to feed the larvae. So one of our most important things in the fermentation process are the yeast and the molds that live on the pollen. And they're important for developing the nutrients that the bees can't get anywhere else in their diet. So if you have pollen that's been sprayed with fungicides, the whole purpose of the fungicide is to shut down the fungal development. So what happens now to that pollen when the bees go to inoculate it? And there is evidence now that at least some fungicides have a very detrimental impact on the fermentation of the pollen and the resultant health of the bees. So that is a big one, and it's concerning too because we have so much emphasis right now on pesticides. When people say pesticides, everything kind of gets lumped into one category. But fungicides are also pesticides, but I think we really need to look at them as a separate chemical and not be using the word pesticide and not understanding that the fungicide is actually doing a very different job. Yeah. Yeah, and then
Emergency.
trying to rear queens when the bee bread's been exposed to fungicides. What was it, 75-25? 80-20? Yeah, when we were at the Tucson Bee Lab, we had an opportunity to see like a giant wall poster from a study that they'd been working on. And they took bees from very intense apple production in Washington State, and they had bees that were exposed to pollen that came from the non-organic farm where there was a lot of fungicide being used. And then there were bees from an organic farm that the pollen hadn't been exposed to the same fungicides. And they found that they only had like a 20% success rate rearing queens from the fungicide pollen as they did from the organic farm pollen. And there was some huge percentage of, what was it, supersedure or building of emergency queens. Yeah.
What they did is they took pollen that had fungicides in it and pollen that didn't.
Wait, Dino, come up so they can hear you. He's the detail guy.
I took photographs of the poster, so I got to study it a little bit afterwards. What they did is they took some pollen that had fungicides in it and pollen that didn't have fungicides in it. And they fed those to two groups of bees, and they removed the queens so that the bees had to raise an emergency queen. And everybody started to raise queens, just like if you've ever removed a queen from a hive or accidentally squashed or lost a queen, they just start raising queens. And what they found was that by the end of the study, that the bees that were exposed to fungicides, only 20% of them were able to actually raise to adulthood an emergency queen. The bees that weren't exposed to fungicides, 80% of them were able to raise that queen to adulthood. So whether or not that has to do with the microbes or some other pathway that the fungicide has with the development of the bee, that's a minor detail point. The important thing is that the fungicide actually had an effect on raising queens and on the bee's ability to raise queens, not on commercial queen rearing necessarily, just on how the colony functions. And of course, there's honeybees and then there's all the other kinds of bees and insects that are also exposed to the fungicides from those same flowers. And it would be hard to imagine that it would be a drastically different outcome for the native pollinators as well. Okay?
Yeah, I guess I should qualify what he said, like not exposed to fungicides, because a lot of times people will say, oh, well, this fungicide is organic. I'm practicing organic agriculture. But if you're shutting down microbes, you're shutting down microbes. It almost doesn't matter like what the source is. And oftentimes organic agriculture will get sprayed even more than conventional agriculture because it's a more labor-intensive way to try to control the insects or the molds or the pathogens. So I'm definitely not trying to say that organic agriculture is bad. Just these things are very complicated, and we can't always assume that just because something is organic means that chemicals aren't being used, and that those chemicals that are being used aren't impacting the food supply. So, again, fallout on the war on microbes, the fungicides may be more harmful than the pesticides, and we're especially seeing this now with huge emphasis now on the neonicotinoids and people insisting that that's at the root of the problem with the bee health. And meanwhile, there's all these other things going on that no one's really aggressively looking at that might actually be much more harmful. And I personally think it's really sad for everybody to be sort of on one bandwagon. We might win the war and get the neonics banished, but what are they going to get replaced with? And meanwhile, there might be other things going on that we're not spending energy trying to figure out that might actually be more helpful in the long run to understand. So we do know that antibiotics can change the climate of the gut microbes and disrupt the workforce in the gut. And again, those microbes in the gut, they're helping the bees to digest and assimilate their food. They're creating antibiotics. If we're holding space against more virulent microbes. And basically, they're at the center of the bees' kind of metabolism and functioning.
And it's not a temporary, it goes generation to generation.
Right, and that's, yeah, I didn't put a slide in here about the antibiotics, but Dean will talk about this a little bit in his talk, right? The antibiotic resistance?
I don't have it in my talk.
You don't have it? Okay. Basically, some of the work that the Moran lab, that Nancy Moran did a couple years ago, was looking at the gut microbes between treated and untreated bees. And bees that hadn't been treated ever with antibiotics, like countries like New Zealand and Switzerland, they don't use antibiotics at all there. Those bees had very, very different microbes in their guts. No, the microbes weren't different, but the resistance that the microbes showing to antibiotics was really different. So bees that had never been exposed to microbes had very, very low level of antibiotic resistance, and that's just sort of built into the microbes to resist other pathogens. The bees that had been exposed to microbes repeatedly showed very high levels of antibiotic resistance. And then the lab also looked at D. Lesby's bees in Arizona, and even though her bees hadn't been treated with antibiotics for more than 25 years, they still showed levels of antibiotic resistance. So these things are, all of us that are getting bees, basically most of us are probably getting bees at least in the beginning or generations from packaged bees. Those bees are pretty much treated prophylactically in the industry. So there's this whole, even if we're not treating with antibiotics, what's happened to our bees' gut microbes over generations, if it takes more than 25 years for the bees to revert almost back to where they were in New Zealand or Switzerland, but are still showing resistance, what's going on with all the rest of the bees? And I'm kind of bringing up things, Kirk sent me an awesome email and asked me so many questions about like, okay, great to hear about the microbes and how important they are, but what are we supposed to do? And I thought about it, and I honestly don't have a lot of answers, because a lot of this stuff, it's sort of, we're now with bees the way we were with human microbes maybe 10 or 20 years ago, trying to understand even the basic stuff and what the impacts are of anything that we do. So we know on some level there is an impact, but how big of an impact, and are the choices that we're making better or worse than making a different choice? I honestly don't know. I feel bad that I don't know. I wish I knew and I could have answers for everybody, but basically what I have now is just a lot of information. Again, the targeted killings can result in collateral damage. We're trying to deal with one issue. Say we're trying to deal with mites in our hive, and we explore all our options, and we decide, okay, I really don't want to treat, but I feel like I have to treat. I'm going to pick this thing. It seems like it's the least virulent thing. I'll pick like Thymol. Well, do we honestly know what the Thymol is doing to the microbes? We know that that Thymol, a tiny little crystal of Thymol in a jar of library paste, can keep that paste from getting moldy indefinitely. It's very powerful. So, but do we know what effect it's having on the bee bread? Do we know what effect it's having on the bee's gut microbes? These are things that we don't know. Like, we know that there's evidence that there is an effect, but in the real world, what is that effect? So the field is more complex than the war room. We have our laboratory studies and we can see that things are happening. But what happens out in the field, and especially in the bee world where the bees are flying freely, there's so many things going on, there's so many variables, that it's really hard to figure out conclusively what's leading to what and what choices we should make. So every decision has a cost. It may not be possible to have a clear understanding of the details or scope of the cost. So again, the honeybee social microbial culture, it uses the simple inputs to provide for the complex needs. It's heritable, persistent, and adaptable. And transmits vertically from generation to generation. So the bees have their own, the microbes have their own biology, and as they're reproducing, they're carrying with them what's happened to them before. And just like in people, the honeybee social microbial culture may be more important to the honeybee health and well-being than genetics. And one thing that, I thought about back, the first talk I ever gave in 2008 on the microbes was in Nebraska. And one of the first things when I was doing some research, I thought, maybe there's something about swapping home. We're always told, like, there's two messages. One is if you have bees that are weak and you have bees that are strong. If you take a frame from the strong bees, young, healthy, brood about ready to emerge and young, healthy nurse bees, and you give that to a weaker hive, you can basically boost the hive. And we're always taught, well, it's on a population level, right? The hive's week, you're gonna contribute like a few thousand young, new, fresh bees. But I thought, what if what you're really transferring is the healthy microbes? What if the reason why that healthy hive is healthy is because their microbes are better? And when you transfer that frame over, you're basically inoculating the hive with healthy microbes. That's something that I think about a lot. But the take-home message, basically across the board, there's an increasing awareness of the importance of the microbes for healthy, well-nourished bees. And the healthy, well-nourished bees are the best defense against disease. So a little, again, Martha Gilliam, when she was doing her research, she identified over 8,000 bee-related microbes, but she was using culturing techniques. They didn't have DNA sequencing back when she was working. So it was unclear, like, which of these microbes were actually, like, the really predominant vital ones and which ones were sort of, like, peripheral. Now with the DNA sequencing, we can understand better the honeybee gut microbial ecology, and that's what Nancy Moran's working on in her lab. And she's basically the only researcher that we know that's been willing to look at treated versus untreated bees. It's a really important thing if you're looking at microbes to be able to look at populations that have not experienced antibiotics and that aren't being treated with other things.
And other antimicrobials like thymol.
Right, and other antimicrobials like thymol or organic acids. So we have a lot of unanswered questions. How exactly do bees become inoculated? This is something that, again, we have so many questions. We were really lucky that we can email Nancy and ask her questions, and she emails back almost immediately. She's very, very generous with her time and her knowledge. But she's working with fundamental stuff. Like, her idea is that she's building a wall, and that wall is brick by brick. We're like up here, we're like, what about the edifice? What about the clock tower? She's like, no, we're down here on the foundation. So some of these answers might take a long time to come, but there's still things that we wonder about. And also, what about queens and drones? Most of the research happens on the worker level. How are the queens getting inoculated? Are they inoculated the same way that the workers are? Do they carry the same gut microbes? How do the microbial cultures affect the fertility? We have so many things in the bee world now about problems with fertility in queens and in drones. What effect might the microbes have in that? Dave Tarpey came to our club, I think in 2010, 2011, and he had a chart showing nosema infections in queens through, like, I don't know, since the 1930s. It's like, 1930, 15% of the queens produced by the producers had nosema. By 1940, it might have been 20%. By 1950, maybe 10%. But percentages all along. And then it was like 2010, like 0% nosema infection in the queens. And we were like, whoa, what happened? Like, why no infection? And he said, because the queen breeders are feeding the snot out of them with fumagillin. It was like, oh. Well, okay, so they're not exhibiting a nosema infection, but like, what's that doing to all the rest of their microbes? And that's something that I don't think anybody really knows right now. Also, drones. If drones are basically half the equation, what's up with the drones? What are their microbes like? I don't know that anybody's looking at drone guts and figuring out how their cultures may be the same or different from the workers. And also, what effect do the drifting drones have on the microbial cultures of hives they visit? So you have a lot of feeding of the drones. Are the drones being fed poop from the workers? Are the drones being cleaned up by the workers? Like, what's going on that level? We don't really know. And another one that seems really big to me that I can find no information on, and we actually have a friend, a researcher down in Georgia, who's a plant specialist. And I asked him, can you find me any literature on the relationship between the microbes that support the roots of the plants and the nutrition of the pollen and nectar that they're offering? And he said he could not find any literature out there. So that's another question I have about, like, the health and vitality of the actual food that the bees are getting, even before it's impacted by pesticides, fungicides, acids. Is there something going on there? That any kind of relationship between health at the root of the plant and quality of the pollen and nectar. So all questions and not a lot of real answers. So some resources. our website, we have a bunch of microbial articles that you can, most of them you can just click and it'll take you to the source. We also have the Gilliam Archives, which is, I don't know, 2 huge binders full of Martha Gilliam's research from the Tucson Bee Lab that we were able to scan and put on our website. Nancy's website is the Bee Microbiome Project. She's in Texas. A book that I'll have available for people to look at. I forgot to bring it on stage, but it's basically a $85, almost like a textbook, but it's scientific articles from around the world on bees and microbes that's been the articles have kind of been rewritten so they're a little more reader-friendly for somebody who's not a scientist just trying to read a scientific study. So they have all of the facts from the scientific studies, but written in a way that's much more interesting to read. And that's a book that I think every bee club should have. It's expensive for an individual to own it, but if you have it as a resource for people to share, the articles are actually really interesting. And then you can Google the Human Microbiome Project and find hundreds of articles on humans and microbes, and I think many of those things that we're finding out about humans will parallel at some point to the bees. So that's it. More, just feel free to come up and say hi. We're very friendly. Oh, I'm sorry. Questions? Yeah. Yes.
I was the one at, you guys are from New England, right? So where's the Yankee ingenuity? Was sort of the question that I emailed you. Sort of what is the how rather than the why? See, and I just want to say you've done a really fantastic job in presenting the big picture and putting it and showing all the pieces and the questions at the end and stuff. That's great. So I think in some ways we just have to understand that this is a conversation, this is just the beginning. And that it's really great that you're building the foundation for us in terms of understanding what the pieces are. And then we can kind of build on that. I think that's wonderful. The one thing to mention, I did see Dean's videos. He has several out, like a talk at Google and stuff. So I watched those, and those are really good. This has been videoed, and I think the video will be available. Somebody can speak to that later. But just to sort of, again, spread the knowledge so that we can generate a much bigger and more rigorous conversation about this. The only practical question I can ask is, I had some frames of pollen that were left over from the hive that died out, and they got really moldy. And I ended up putting it back in the hive to see how they would handle, basically let them clean out the comb and reuse it. And they managed to survive. I don't know what I did when I put that back in there, but do you have any thoughts on that? It's just a very simple, quick question.
What color was the mold?
It was kind of green and moldy.
Yeah, I've heard people, everybody I know will say, like, if it's green mold, don't worry about it. Some people say if it's black mold, like, not great, but the bees do a really good job at cleaning that up.
Especially a strong colony.
Yeah, especially a strong colony. I wouldn't give moldy pollen to a weak colony. But I wouldn't give a lot of things to a weak colony. It's like a strong, healthy person can take on a lot more than someone with a weakened immune system. But, yeah, then I forgot, wait, just a minute. You reminded me that we had the conversation in the lobby, but I forgot to talk here about, we talked about the poop. Has anyone here heard about the human fecal transplant? Okay, so that's, to me, a really big parallel. People that have had to undergo intravenous antibiotics often have their gut microbes just totally knocked down, and the space that the healthy microbes were holding, or the space that the microbes that we want to have in there were holding is suddenly like empty, and a virulent microbe can move in. And there's a microbe called C. difficile that can basically kill you. And for a long time, people that had that suffered horribly. Adult people, they had to wear diapers, they couldn't go out, their gut was just completely non-functional. And a Danish doctor, I don't know if it was back around 2005 or 2007, he started doing fecal transplants using poop from a healthy donor mixed with some distilled water, a tube down the nose and throat of the affected individual, bypassing the stomach acids, because the stomach is a very acidic environment and would have been too hostile for the bacteria. Get the tube down into the intestine and basically inoculate the person with the healthy poop. And after 2 or 3 treatments, they have like a 95% success rate, which in medicine is really big. Like 95%. They were failed patients. Like, there was nothing to do for these people. They were literally hopeless. And with the fecal transplant, they were all brought back to health, 95% of them. So to me, that's like a really, and now we have people that are saying, well, what are those specific microbes and how can we make them into a pill? And you know, the thing itself is whole and works. And I don't know if it in my mind, like, why would you want to treat it as a commodity and turn it into like a product when like the real thing works? But that's how I guess medicine works. You know, you have an industry that wants to have a product that it can sell, and people that want to have a pill they can swallow that isn't as gross to think about as like a poop infusion. But that to me was one of the most powerful things that I've read. And there's so many interesting stories. The New York Times had a fascinating article. It was an editorial written by a woman who was a donor for a friend who almost died. And that article had a lot of really interesting things in it about the whole history of the fecal inoculation and etc. So anyway, I would ask any of you to Google that, and you can learn a lot from that one article.
So,
Kirk sent us this email, and you know, there's different kinds of email that you get, and when you get an email from somebody that's actually watched, you know, read your book and watched some of your videos and given it some thought and asked some questions, that's different than somebody that just wants an easy answer, what should I do, without doing any work on their own. So, you know, we consider that very, like, we're successful if we're getting that feedback, and especially if we don't even have a good answer for it. But there's a couple of things that I think are worth talking about. First of all, this thing where these microbial communities hold the environment and occupy a space and keep other things from colonizing, I think is really one of the most important things. Our focus is on keeping bees without treatments. Now, and I think the gut microbes and the bees in this country are compromised. Even D. lesbies bees, you know, we can see in a lab study that they're not what they are in countries that have never seen antibiotics, even though they haven't seen antibiotics in 26 years plus. So, you know, there's an important point that I think that if you're going to keep bees without treatments, or if you're going to have healthy bees, and when I say, you know, are your bees healthy, and you say, yes, my bees are healthy because I give them mite treatments and antibiotics every year, you know, I don't know, that doesn't sound so healthy to me if you think that your bees are one season away from dying if you take away these very strong medications that have a big impact on the bees and on their microbes. But I think the key is for all of us, and I don't necessarily even mean as individual beekeepers, I mean as beekeepers, to keep the bees alive long enough to get those populations to stabilize. Ramona talked about how these populations, these cultures are always balancing, but they're never balanced. You know, it isn't like we're going to get, okay, now we're balanced and we're done. It's always pushing a little bit this way, and you know, even, you know, something happens and it's pushing a little bit that way, and that ability for that culture to keep itself in balance and for none of the individual components to take over, because if any individual component of that takes over, they destroy the environment not only for their competition but for themselves, because it's the waste from the other members of that community that's providing the food for them and is providing the right environment for them. So I think that's really the goal, is to maintain these cultures long enough that they can stabilize to a point where they can support the bees, because that's what they do. That's what they've always done. And clearly they're capable of it, and mites and nosema, these are not the first, you know, new challenges that bees have faced, and yet they're still here. So obviously we've stuck our finger in the pie here and you know, put, you know, pesticides in the environment and fungicides, and it's more complicated. But when we look at these systems, I think keeping them alive long enough to stabilize is the important part. But there's a flip side to that. You know, I'm not going to tell you that, you know, have we lost 90% of our bees by not treating? Yes, we have. At times, some years, yes. Some years we've had 90% survival. It swings either way. We've made a decision that we're going to try to keep enough bees and to try to keep this thing going for us. But the problem is, if you've got 2 hives, and you might lose 90% of them if you don't treat them, you don't have 2 hives in the spring. That's a problem. And we get people all the time, and I had this conversation just before we started talking this morning with somebody. We get people all the time that say, well, is it okay if I do this? Or, well, I only have one hive and they seem to be doing okay, but it's the fall and the mite levels are building up, and I really think, I don't think that they're going to make it if I don't do something. Is it okay if I use X, or is it okay if I use Y? I'm not your judge or your boss or your apiary inspector. You have to feel comfortable with what you're doing for yourself and for your bees. And as Ramona said, that everything has a cost. Not treating has a cost. Now, it also has a benefit. Treating has a cost, and it also has a benefit. And whether you're going to use, antibiotics or essential oils, those costs and benefits change. One kind of misinterpretation that I think people have is that these natural products, that these soft treatments are better and more natural, and therefore going to not cause as much harm. But remember, something like Thymol. First of all, the Thymol that we put in our hives is not a natural product, it's synthesized. It's not fields of thyme being grown and distilled. But that doesn't mean it's not the same substance. It doesn't mean it's not a naturally occurring substance. But in that kind of concentration that we put it into the hive, that's so metabolically expensive that nature would never do it. We don't have essential oils that smell like a bottle of thyme, like a bottle of Thymol, or like a Thymol patty. And we have to understand that it may be that something like fluvalinate maybe has all these impacts and it's bad for the bees and does all these things, but it probably doesn't have as much of an impact on the microbial communities as some of these natural treatments, because thyme makes Thymol to keep the bacteria and other microorganisms and deer and other predators from eating it. That's what we love maple syrup. We love maple syrup. It tastes terrific. But that thing that's the taste, those are the toxins that the maple tree is producing to keep the microbes and the fungi from colonizing the tree while it's alive.
Also the syrup is like highly concentrated.
Right, and the syrup is, I forget what the ratio, a 40th of, you're boiling off 40 parts water to get 1 part syrup. Nature would never do that because it's too expensive. And so we're introducing these things that seem natural, but we're putting them in forms that they just don't otherwise exist. There's uranium ore out there in the world, and for the most part, people have been able to exist with that without huge health effects. But when we concentrate it into uranium rods and use it in a nuclear power plant, then we have to figure out something to do with them so that they don't destroy the planet that we're a part of. So, and the example that we use in our book is, would you want a natural massage oil? Great. How about a natural massage oil made from poison ivy oil? That's not such a good idea. For somebody it might be a good idea. I've got a list of people that would be good for. But just because it's natural or just because it seems natural doesn't mean that it's fostering the microbial community or fostering the bees more than something that might be synthetic. So those were my 2 thoughts on that.
Thank you. Anybody else? Yeah?
Did you raise your hand? Everyone set? Thank you.